Hi, I'm Kert Viele. I'm a director and senior statistical scientist with Berry Consultants, and I'd like to talk to you today about the use of historical information, historical borrowing in clinical trials. Typically when we run a trial, we have a control arm. We're comparing against a novel treatment. That control arm it rarely exists in a vacuum. It's usually been in a number of prior studies. The studies that were used for approval, the study that were made post-approval. Perhaps that drug is now the standard of care, and other kinds of studies have been done against other novel treatments. You have all this information together, you have some idea about what is going on with that control arm, what its rate is, what its mean is, what would be its time of survival. So the question is, can you use that information and figure out what's going on with the control arm and augment your current study with that historical information and therefore be able to do a more efficient trial today? The good part about this is if the previous studies are on point, so if you're running a study and what's been done in the past is applicable, that information is valuable. So we can get more precise estimates. We can eliminate possible biases. Generally what's going to happen is we'll have a better idea of the control arm with the historical studies than without. So that will allow us to make better decisions. We get better point estimates. We get better go, no-go decisions. We get a better idea about whether our drug works. This allows us to potentially eliminate some of the controls in our current study. So we can get trials that are 20% shorter. We can get trials that focus more on the treatment. So being able to use 2-to-1 or 3-to-1 randomization is huge. That would allow us potentially, even in a smaller study, to have more of our subjects on the treatment arm. That lets you assess safety better than you would without using the historical controls. The problem is that if you borrow from an inappropriate source, so there's been some kind of drift over time, the control arm doesn't behave today like it did in the past, what can happen is you can create a bias. So you can say in the past the control rate was 70%, today it might only be 60%, and you generate that bias. The control gets pulled up to the historical data, and that bias is in a bad direction. That gives you the possibility of decreased power. It also gives you the possibility of an increased type 1 error, which is the real regulatory concern with using historical data. So what I want to talk about for the remainder of the talk is how do we get the most of the good points and how do we minimize the bad points here. So the first thing we need to do is we need to get an idea of how much to weight those previous studies. So should we use heavy weight? If we had a lot of historical data and we knew that data was going to be completely on point, we'd want to weight it heavily. It's basically telling us exactly what we want to know. On the other hand, if that historical data is less applicable, then we'd be really worried about the downside. We'd want to downweight it. The extreme of that is not using it at all. That would give it a weight of 0. So the process here, first thing we need to do is we have to get data from the past that we think is applicable. Typically that would happen through previous clinical trials, so you do some kind of literature search. Certainly don't want to leave out studies. You want to worry about publication bias, so make sure this is especially good if it's a drug that's in-house. You may have a lot more historical data than others. You can also find records at the sites where you're going to go. So if your trial is going to happen in a year, you could start now and go to the sites and say, look, we want to compare to standard of care today. So start collecting data and give us an idea about what the control group's going to look like. It's not as good as the controls that happen at the same time as the treatments, but it's information that we can use to make better decisions in the future. Another point that I want to make is a big thing that's happening right now is platform trials. Trials where multiple drugs are being compared and drugs move in and out of the study. So for example, the trial may run for 10 years and you may see new drugs come in, you may see the standard of care improve over time. In those types of settings, you've had a single trial that's been going on for years. So you have control data for all of those years, and not only is it control data on the same drug, it was done at the same sites with the same inclusion-exclusion criteria. That makes it very applicable for this kind of historical borrowing. To get an idea about how this works, I want to start with a simple example. So we have a dichotomous endpoint. Everything's yes or no, success or failure. The current trial has 200 subjects on each of a control and a treatment arm. What I want to do is I want to make use of the fact that in addition to the data in the study, I also have an additional 100 subjects. Maybe this was a POC study that I did. Maybe this was another trial at another location. Maybe this was the data that you got at the sites for the year before you started the trial. But I know that I have 100 subjects and 65 of them were successful. I want to put those together and I want to assess a primary analysis which just says, is my treatment better than the control? I want to get a high rate. So you can see in the graph, what I've got is in the control arm, I've got two pieces. I've got the current subjects, I've got the 65 out of 100 historical subjects. In the treatment arm, I just have my treatment. So I'm trying to augment the control. I am not running a single arm study. I'm trying to combine the historical data with my current data. So the key aspect is how much do I weight? I'm going to start and I'm going to talk about fixed weighting, where you decide in advance how much weight you're going to give the data. What we're going to find is there's some problems with this, but it's important to get this as the baseline. And then when we go forward, we're going to talk about how can we select the weight based on accruing data. This is called dynamic weighting or dynamic borrowing. So we're going to start with the static, we're going to show what the pros and cons are, and then we're going to show how the dynamic allows us to make the most of the benefits and avoid the downsides. So the weighting here, what you can see in the red text, this is saying that every current subject gets a full weight of 1, so 100%. The question is, what is the relative weight that you give to history? So you could give it a weight of 0. So this is what's commonly done if you ignore history. W equals 0, pretend it doesn't exist. You could say W is equal to 1. So you give it the same weight as the current subjects. So that says a subject in the past counts exactly the same as a patient that's now. Those are usually the extremes. Usually you try to pick something in between. The notion is that patients in the past, they probably are not completely applicable, so they're meaningful. But we may only want to give them half weight or a third weight or a fifth weight compared to a patient in the study. We want to take the information in, but we don't want it to swamp what we have now. There are situations where you could overweight. You could say a patient in the past has more weight than a patient at present. And that sounds very unintuitive, and well, it is. It's not the best thing to do, but it's also common for people to run single-arm trials where they don't have a treatment control arm at all. And when you don't have that control arm in the current study, the only thing you have is the patients in the past. And that corresponds to overweighting. And what we'll show is that's a really bad idea. So if you're ever considering a single arm trial, you're trying to say, look, we need to really make this efficient. I want to minimize the number of subjects. A single arm trial doesn't do what you want, but the historical borrowing can let you get most of the benefits, but you still get the advantage of control subjects in the current trial. So with a fixed weight, you would decide in advance, I want to use W equal to 0, W equal to 1, W equal to 0.5, 0.2, 1/5, 1/2, whatever you've picked. You would combine that, the equations here, you would combine the binomial likelihood for the current subjects, combine that with the likelihood for the subjects in the past. So if you had W equal to 0.2, that would be 1/5 weight. So you'd say the 65 out of 100 from the past, divide that by 5, it looks like 13 out of 20 subjects. So you'd be taking the current subjects and acting like you had 20 more where 13 of those were responses. So I'm going to talk about what happens if you do this for W equals 0, ignore history, up to W equal to 1 where you pool, and then I'll also show what happens for a single arm trial. I'm going to do this in the context of the operating characteristics of the trial. I'm going to talk about the point estimates, the mean square error. I'm also going to talk about this in the context of hypothesis testing. So it's very important we make decisions. We want to know what the drug does in terms of the rate, but what we're often really interested in is the treatment better than the control? So we need to talk about in terms of that decision, is it made better with historical borrowing or are there problems? The key aspect of this is an idea called drift. So you've got your historical data. In our example, that's 65% rate. Our current data, we don't know yet. So we're about to design the trial, we're walking into it. But there's a parameter underneath it. I'm going to call that PC. That's our current control rate. Ideally, that's 65% too. That says our historical data was perfectly on point, randomly ended up in the right place. That's going to give us the most advantageous situation. But it's also most likely that our current control rate, PC, is going to either be a little bit higher than 65%, it's going to be a little bit lower than 65%. There's also the possibility it could be very different than 65%. Whatever that difference is, that's drift. It's the difference between the parameter I have now when I'm going into the study and whatever that historical data is that I'm borrowing from. We don't know this in advance. So what we need to do is we need to have a trial that's robust. I can't say in advance, the drift is 0, so I should borrow heavily. I can't say in advance the drift is large, so I should get rid of the historical data. I'm going to need a method that takes the drift into account as I'm doing the study. So for fixed weights, this is a graph of what happens to the mean square error. So you can see lots of curves here. The axes, I've got the drift on the x-axis. So at the bottom, I've got the true control proportion. In the middle is the 65% for history. But the true control proportion, it might be higher, might be 70, 75, might be lower, might be 60, might be 55. So drift is really being measured on the x-axis. On the y-axis, I've got the mean square error. So basically, how far away are our estimates from the truth? And you can see the bottom of that is 0, so I don't make any error at all. And you can see, of course, in any kind of random setting, you encounter some errors. The orange line, so you can see in the middle of that, the orange line, it is flat, so drift doesn't matter much to it. If you ignore history, then it doesn't matter whether the rate is 55, 60, 65, 70, 75. The error rate stays about the same for mean square error. For the red line, that's the W equal to 1. Red corresponds to pooling. I count history identical to the current data. What happens with that is if there's no drift, so if historically I got 65% and my current parameter is 65%, then pooling everything together, this is great. I minimize the variance, I don't create any biases, and you can see I've pulled down that red curve in comparison to ignoring history. The red curve is lower than the orange. You can also see the blue curves, which essentially interpolate. So if I go 20% weight, 40% weight, 60, 80% weight, what happens is I interpolate between that orange and the red. So you get to pick how much you want to weight the historical data. You can see that the range where borrowing beats the orange curve, it depends on the weight. So the less that you borrow, you get less of a benefit, but you get it over a broader range. So if you think, hey, there may be a decent amount of drift, maybe I want to weight this 20 or 40, I get some benefit and I get it over a broad range. I don't have to worry quite what the drift is. I will get a little bit of benefit from doing the borrowing. The purple line is the problematic one. That corresponds to giving infinite weight to the past. This corresponds to a single arm trial. So if I don't use any control data at all in my current trial and just compare to 65, you can see if there's no drift at all, this is fantastic. I do perfectly. On the other hand, if there's any drift whatsoever, this essentially asymptotes. So this is a disaster. So you can imagine W equals 0 is okay, W equals 0.2, 0.4, 0.6, 0.8. There are some risks and benefits. The purple line says I'm either going to win like crazy or I'm going to have a disastrous loss. So generally speaking, this is why we don't like single-arm trials, because they take too much of a risk. Using the blue, or at worst the red, allows you to get some benefit to the historical data, but you're not overweighting it. You're making sure the current controls are there and they can help you assess what the drift is and make the right decision. In terms of the type 1 error and power, so you've got two graphs that you're seeing in front of you. The same color scheme is used. Orange is ignoring history, the blue are reasonable weights, red is pooling, and purple is the single-arm trial. On the left side, you see the type 1 error. On the right side, you see the power. I've powered this for a 12% increase. What you can see is, again, with no drift, things do well. So there's a sweet spot here. You can also see if there's a large upward drift, you can see how you can get into trouble. So the historical data was 65%. Suppose our current data is 65 or 75%. What's going to happen is that historical data, you can imagine if you're doing a high jump. So the bar is at 65%. The historical data, it's pulling that down and it's saying, let's make it to be easier to win. That makes it easier to win. It increases your power. So that's what you see on the right panel. The power is increased meaningfully. It's increased by 10, 20% potentially. But on the right side, you also see the inflated type 1 error. So the FDA would have serious concerns about, say, the red or in particular the purple curves because the inflation is dramatic. So you want to make sure that you pick a weight that doesn't inflate type 1 error too much. In the middle of this curve, what you see is actually the type 1 error is decreased. So the blue curves are actually underneath the orange, or equal to it. Whereas for the power curve, you get more power. This is where we're allowed to get 20% fewer subjects into our study and still achieve comparable power. All of this is aimed at an assumption that the drift is not going to be too much. If the drift is too high, we're going to run into type 1 error concerns. If the drift is too low, on the low side, we're going to end up with type 1 error concerns. We want to essentially be Goldilocks. We want to end up with a modest amount of drift. Doesn't have to be zero, but a modest amount of drift. And then we can get big benefits from this. So a key point here, if we knew the drift, we'd know how much to weight. If we knew the drift was zero, we'd weight heavily. If we knew the drift was large, we might ignore history. We'd weight very low. We never know this in advance. So do we pick W equal to 0.2? Do we pick 0.5? Do we pick 0.8? We don't know before the study. But we do find out during the study how close it is. If I went into the study and I had a 65% historical rate, if I'm looking at my data and the data's coming in and it's about, say, 63, 67, 68, anywhere in the 60s really, I go, look, that's close. That's a situation I want to weight heavily. If my current data comes in, remember my history was at 65, my current data comes in at 80 or my current data comes in at 50, that says, uh-oh, whatever was happening in the past is not happening now. So I don't want to use that historical data anymore. So what we want is a method that when the drift appears to be small, we're going to give high weight. We're going to get the most of the benefits. But when the drift appears to be large, large up, large down, what we're going to do is we're then going to suddenly discount history. We're not going to use it anymore. This all goes under the notion of dynamic borrowing. Want to make sure that we adjust the weight depending on what's happening in the current study. The way we do this is hierarchical models. So the PC that we talked about earlier, that's the current control rate. In the historical studies, there are also control rates. We're going to call them P1, P2, up to PH. The H is for history. We're going to put all of those together. So you can see at the bottom the logit of PC, the logit of P1, the logit of P2. We're going to assume that those all come from a normal distribution. Doesn't mean they're all the same, but it means there's some pattern of variation. If I repeat a study, if I examine the same control arm, I do it today, I did it a year ago, I did it 2 years ago, all of these studies have some natural pattern of variation. So that's described by a mean mu, it's described by a standard deviation tau. I'm also going to put priors on those. So I'm going to have to estimate mu, I'm going to have to estimate tau. The key parameter here is the tau. So that's measuring your across study variation. If tau is very, very small, what you're saying is study to study, I get the same results. So when tau is small, you're saying I'm not going to have much drift. When tau is large, that says study to study, anything can happen. So that says I may have large drift. If anything can happen, I'm fairly likely I got 65% before, but now I can get anything. I can get 80, I can get 50. So when tau is large, that's a situation that's bad. If you fix a weight, that's equivalent to fixing a tau. So what we're going to try to do, we put a prior on tau, we're going to try to estimate that given the data. So we're going to come in, compute our posterior distribution. We get a posterior distribution on tau. Gives us an idea, is tau big, is tau small? And then we're going to pick a weight corresponding to that. This generates what's called dynamic borrowing. And the graph that you can see here, again, I have the drift on the x-axis, what I'm trying to show throughout the talk. On the y-axis here, I have an effective number of borrowed subjects. So you can think about a weight. If I weighted the historical data, I had 100 subjects. If I weight it fully, then I'm going to borrow the equivalent of 100 subjects. If I have a weight of 0.2, what I'm considering is that 65 out of 100, I'm treating it as 13 out of 20. So that's the equivalent of only borrowing 20 subjects. So the y-axis is saying what is the effective number of borrowed subjects. If we were picking a fixed weight in advance, we're saying no matter what the drift is, I'm always going to borrow the equivalent of 20 subjects. I'm always going to borrow the equivalent of 40. With dynamic borrowing, you're trying to say if the drift is small, so you can see if we're at 65% in those graphs, I'm going to borrow the most. All of those curves have a peak. So it might be at 20, it might be at 40. You can set where that peak is. You can set the maximum. So you can say, I'm going to borrow the most when the drift is small, but I never want to borrow more than 20 subjects, never want to borrow more than 40. But the key thing about dynamic borrowing is as drift is encountered, you get drift to the high side, drift to the low side, it stops borrowing. So when the drift starts to hit 80 or the drift starts to get 50, you can see you're not borrowing any subjects anymore. The next topic is to go back and talk about the mean square error for dynamic borrowing. So remember the curves that we had before, they're not in boldface anymore, but we had blue curves, red curves, purple curves, and they all had this property that the borrowing, the mean square error, it inflated. So as the drift increased. The key aspect of the dynamic borrowing is that inflation, it's bounded. If you look at that curve, you can see it does better than the orange, it does better than not borrowing, but what also happens is it's bounded. The green curve, it goes up, it starts to inflate a little bit, but then it goes, hey, hold on a second, this isn't right. So I've got too much drift, let's stop borrowing, and those curves go down. This is a way of bounding your risk. So it's a way of making sure that bad things don't happen. You also get this for type 1 error in power. So you can see, for example, the type 1 error is bounded. So that inflation, the inflation that you get from a high weight, the inflation you get from pooling, the inflation you get from a single arm trial is enormous. I don't even have it shown here anymore. But the inflation from dynamic borrowing, it's bounded, you're bounding your risks. You can say it will never get worse than X, and you have control over what X is. At the same time, you retain your power advantages. So that 20% savings on subjects, you get this with the dynamic borrowing. So as long as things are going well, now if you get a lot of drift, then you're going to have to increase the sample size. But if the drift is what you anticipate, you get the benefits. You keep those with dynamic borrowing, even compared to static. So an example here, Antibiotic trial. This is a very popular topic to consider these days. Lots of literature on this. Original design, think of a non-inferiority trial. A trial that requires 750 subjects with 375 per arm. Remember, you'd have to do 2 of these. So this would be 1,500 subjects in total. With historical borrowing, what you can do is you can change this. So instead of needing the 750, what you need is 600. And so that's 20% fewer. You can also, because the savings, the information that you're bringing in, it's on the control arm, it makes sense to reduce the control arm and not to reduce the treatment. So you would typically do 2-to-1 randomization, for example. That's 200 to 400. So even though I have fewer subjects, what I've done is I actually have more subjects on treatment with this design than I did in the original. So for expected drift, the advantages, remember the prior graphs, you've bounded the risks, the control of type 1 error is still there, and you have comparable power. So you get smaller sample size, more subjects on treatment, and equivalent decisions. So generally speaking, whenever we synthesize data, things can go either direction. The more information that you bring into a trial, you can always make better decisions. What we're trying to do here is incorrect information. It can certainly pull estimates in the wrong direction. The dynamic borrowing is trying to protect that. So we're trying to get this idea of where are we going to improve. We never want to reinvent the wheel. If the control data was applicable before, we should use it. Dynamic borrowing allows us to do that. On the other hand, we also want to be robust. So if new things are coming in, standard of care has changed. In antibiotics, suppose that there's been a development of resistance, that might change things from the past. We want a method that gets away from the borrowing and is robust. Try to get the best of both worlds. So in summary, historical borrowing, it allows us to improve our point estimates. We get lower mean square error. It also allows us to actually reduce type 1 error. So as long as the drift is small, we reduce the variance. It reduces the type 1 error. We can increase the power. That allows us, if we've increased the power with equivalent sample sizes, what's typically done, of course, is we trade that. So we say, let's get the same power, but let's reduce the sample size. Let's run a smaller trial that can do equivalently well. There are situations where historical borrowing is not appropriate. So if you expect a large amount of drift in advance, you don't want to do this. So if you think, well, it's quite possible I could get 80 or it might get 50, then don't do it. So pick a trial that realizes that you're in a new reality. However, if you have a trial where you think that you have a small amount of drift, but you're worried that you might have a problem, dynamic borrowing allows you to take the advantages of the small amount of drift and limit your risks, and you don't have to face the downside. So I hope you are successful in making your trials more efficient. Thank you very much.